Ladies and gentlemen of the general public, members of the medical and scientific community, we're here today and for the next several shows to ask a series of scientific and business experts about the world of stem cells. We're going to be looking at the science, the medical uses, and the technology deployed in this exciting and fast-moving field. In this episode, we're going to take a deeper look at the difference between autologous stem cells for the individual and allogeneic stem cells off the shelf. Our experts are going to talk about the differences in IP treatment, research, medical application, and the financeability of these different approaches. Some of the major questions we're going to deal with in this particular episode are, what are the exact differences scientifically between autologous and allogeneic stem cells, and where do they come from? What is the FDA's attitude toward the treatment on a regulatory level of autologous or allogeneic stem cells? Is there a difference in IP treatment as well? And how are these stem cells, autologous and allogeneic, deployed differently?
If you take a cell from the patient and then return it after you've done something to it, you've expanded it or modified, you put it back in the patient, it's the patient's own cells going back. Number one is there's an attitude by the regulatory bodies that is a much more acceptable thing. First of all, you're not necessarily influencing the immune system, which is a very, very powerful player in any transplantation procedure. So you're not going to get a marked rejection phenomenon if you return the patient's own cells. So that's risk has been reduced that you get an adverse event due to a difference in the immunology of the cells that you're putting in and the patient. When it comes to allogenic cells, so you've got an opportunity to put the cells in a vial, send it out, distribute it basically like a drug. And if you can utilize those cells to treat serious conditions, say like diabetes, type 1, type 2 diabetes, and you can get cells in a protected environment that is not susceptible to the immune system, if you can protect them in a capsule of some kind, where they can still respond to the glucose levels and also still produce the insulin that you need in a robust manner to maintain you in a steady state. And you can protect it from the immune system. You've got a functional entity that will allow you to work with allogenic cells. Or if you put the cells in a place, maybe the eye is a protected environment, essentially a privileged site, and you're probably not going to get a lot of rejection going on in the eye because it's essentially privileged from the attack of the immune system.
It's not easy to see what that business model is, neither for autologous nor allogeneic therapy. But we do have examples of autologous therapy which has entered the market with things like Carticel from Genzyme. And my understanding is these things do not make a lot of money, do not continuously excite the investors, but they act as a proof of principle that you can get a cell therapy product to the market. I think everyone would agree that if you can get an allogeneic product made and you can conquer the immune rejection issues, then that would be far better starting material for a company.
There are strengths and weaknesses with each of those technologies. The allogeneic cell, or cells that are designed to be pills in a bottle, that you would create a lot of cells and then use them, create a batch of cells, then use them for a whole host of different patients, presents a business model that is more familiar to us because it looks more like the pills in the bottle, big pharma model. And so on some respects, that is an easier investment proposition because you've got a business model that people are familiar with, and some of the paths to successful commercialization, so at least some of the road bumps, the speed bumps along the path to successful commercialization are well known and solutions are there.
Because so many companies are interested in both autologous and allogeneic, the IP landscape is a very troubled one. And it's been troubled because there are some early pioneers that have held on to deep positions in either allogeneic or the autologous stem cell side. We have some experts who are going to help us clarify that.
Early patents in the embryonic space, for instance, there are two major patent portfolios there. One comes out of UMass and the other comes out of Jamie Thompson's lab at University of Madison, Wisconsin. Wharf was the primary beneficiary of those patents, and of course they licensed those patents to Geron. And Wharf and Geron both took a very aggressive view of patent enforcement and patent infringement and of their rights within those patents. And that view had the effect, I think, of limiting activity in new companies and folks who were interested in trying to pursue embryonic stem cell technologies because they were facing such a large patent wall and a concern that they would not be able to work effectively within that context. So I think early on that patent portfolio and the strategy of enforcement around those patents did tend to limit some of the research that was being done in the embryonic space.
What people are remembering was the very dominant IP position that Y-Cell had and licensed to Geron around human embryonic stem cells. And I believe, because I was in a sense victimized by it, that they were very aggressive in their negotiations, and it actually put off many institutions from receiving the cells. Indeed, when we first appealed to Y-Cell to receive human embryonic stem cells, the material transfer agreements were so restrictive and onerous that my institution would not sign them. And what that did was that really hindered the greater dissemination of these research tools and slowed the whole field.
Well, our policy is to make this technology available to everybody. So we don't want to make any limitation or restriction on the usage of this technology. So that's our policy. And that's why Kyoto University has now have a company called Academia Japan, IPSL Academia Japan, which handles all the IP, intellectual property, about iPSL technology.
Time heals all wounds, right? So those patents are going to come off. The Wharf patents, the enabling Wharf patents expire in 3-4 years. So that obstacle to pursuing work in the embryonic area will dissipate. Those obstacles will dissipate over time. In the early days, as the PTO was beginning to understand the technology, they were climbing a learning curve and I don't think fully appreciated what they were working with. And as a result, they granted patents that now in hindsight appear to be competing. And part of that problem stems from how patents were granted, what it took to identify a unique tissue type that you might be able to get intellectual property patent protection around. In the early days, folks were looking at cell surface markers with the understanding or the thought that if you had only 3 or 4 unique cell surface markers, you had a unique cell, and therefore you could patent that cell if you could isolate it and purify it and expand it. And it turns out that you had a little bit of the guy pulling on the trunk of the elephant and the guy pulling on the tail of the elephant, both thinking they have different animals when in fact they're both holding onto the elephant. An example is there is a company that, again, you know well, Osiris. Osiris has got a large patent estate around mesenchymal stem cells based on 3 or 4 specific cell surface markers, and it turns out that there are others who have filed on cells that functionally look very much like the mesenchymal stem cells that Osiris has patented, but they're identified by different cell surface markers. And so you're set up, you have these patents that are just on a path to a collision course. And that is, of course, going to create some intellectual property battles, and the folks who benefit from that will be the intellectual property lawyers representing the parties.
IPS, I think, is different. There is some important early intellectual property around iPS cells. It'll be years before it's ever worked out.
Initial patent has been approved in Japan, but at the moment only in Japan. We are not sure about in the States or in any other countries. And of course, there are many patents are required to realize the application of this technology, iPS cell technology. Any single patent is not sufficient.
But I think that there's enough freedom to operate with different mechanisms of reprogramming that we're going to use that platform. I also think there's appreciation in the scientific community and probably the venture community as well, that this platform needs to be exploited as widely as possible. And that what we're really going to want to protect from an IP perspective is the composition of matter of the molecules that are discovered using this technique. Those are defensible patents. And I think the whole field is going to use the tools the way they should in a relatively non-exclusive manner and get as quickly as possible to patentable, protectable, but importantly, medically valuable drugs.
Regulatory authorities around the world, the FDA in particular, have very different ideas about immunogenicity and self-treatment with your own cells. And therefore, the autologous cell treatment approach has a totally different regulatory regime than allogeneic, which needs regulations to deal with release batches and controls and consistency and stability. So our experts today are going to come forward and give you their views about the evolving regulatory approaches by the FDA and overseas governments on this all-important piece of legislation and statutory control over the stem cell world.
On one hand, a patient who's having autologous cell therapies has to remain as an individual patient under medical care in a hospital that has a clinician who's interested in that particular condition. So you have to find the right doctor to work in that system. Whereas if you have allogenic cells, which are addressing major conditions like, say, type 2 diabetes, where there's a large number of patients, you want it to be in an outpatient scenario, relatively low cost, high efficacy, and the ability to be able to visit the appropriate clinic that provides those kind of treatments more broadly. So yes, indeed, there would be a very different aspect to the way it's delivered. Well, the approval path will be different as well, because transplantation of tissue transplantation tends to be in a category all of its own. And I think once you move into allogenic cells, you've now got a product which is much more like a drug product that's got to conform very much to the requirements of the registration of a particular drug. So you've got to prove that your batches are exactly the same and that you've got the appropriate quality control systems built in, so that when a patient gets this treatment, they're going to respond in a predictable fashion as they do to drugs. So in the case of autologous medicines, you don't really have that same requirement. You're able to get the delivery through the medical system. So they operate in really quite a different framework.
I think FDA has been quite open in my experience for understanding cell-based therapies and being open to demonstrating from the applicant for the clinical trial that there are safe and effective measures that can be taken for manufacturing and for delivering a highly standardized product. In many cases, it is a biological that's defined by the manufacturing process, by the isolation process, and not by strictly a chemical composition.
There have been lots of cells that have been cultured and taken through the FDA. I think the FDA is pretty comfortable doing that. But with these very primitive stem cells, it opens up new issues that I think there's a lot of unknowns about. These are cells that are going to persist in the body, ideally for the life of the individual. So the issue of how long we have to follow up to understand the incidence of adverse events is an open question. These are new entities. You know, we've had cartilage cells and skin cells and various corneal cells and all that have been cultured, manipulated, mesenchymal stem cells and the like. There's a fair amount of experience with that. But taking cells whose origin was in a pluripotent cell, that's really opens up whole new challenges.
For adult stem cells, where we have a greater history, if you think about it, we've been doing adult stem cells for over 40 years. A bone marrow transplant is an adult stem cell treatment, and we've been doing bone marrow transplants for 40 years. So there's a larger body of knowledge. The risk factors associated with adult stem cells are better known, and therefore the regulatory pathway is a little better established. For embryonic cells, the regulatory pathway is being worked out. And one of the leading proponents of that regulatory pathway, or the advanced guard in that regulatory pathway, as it were, is Geron. Geron has been trying to get into the clinic with their therapy derived from human embryonic stem cells for spinal cord for a number of years, and they've just faced a number of challenges. And the FDA is learning about the risk profiles associated with embryonic stem cells and what are appropriate measures that should be taken in a clinical format to address those risk profiles. For embryonic cells, the primary issue that is driving them right now is safety. And the issue with safety is that embryonic stem cells, both the challenge and the weakness of embryonic stem cells is that these cells have the incredible capacity to differentiate into every cell type in the body. And honestly, one of the ways that you know you have an embryonic stem cell is if you take it and put it into an animal model, you will see a teratoma form. And a teratoma is a large tissue mass. It's benign, but it represents most of the tissue types in the body. And in fact, that's one of the defining characteristics of an embryonic stem cell. Well, if you're looking at a therapeutic setting, the last thing you want to do is grow a teratoma. And since embryonic stem cells have that capability, the FDA is very rightly concerned about ensuring that you do not trigger unwanted tissue growth by introduction of cell therapies derived from embryonic stem cells. So they have a very legitimate concern about safety, and they're simply trying to work it out right now. Now, the good news is that Geron has now been taken off clinical hold. It's my understanding they're going to begin their clinical program at some point this fall, and there are others coming up behind them. Novacell has a program in diabetes, and there are other companies that are looking at bringing embryonic stem cell therapies into the market. There's an RPE program that Pfizer and the Peter Coffey and the folks at UCL are working on. ACT's got an RPE program as well. So there are a number of programs that are in the pipeline that eventually be working their way through the clinic. And the more of these clinical programs that get approved, the more comfortable the FDA will become with this technology, and the better they will understand the safety risk profile and how to manage it.
The interesting thing about Europe is that it does actually have this kind of EMEA-driven advanced therapies regulations now, which have now come into effect as of the end of last year. And they do allow, I mean, from a marketing perspective, once you get a product that you can then market, it's a long way off from where we are now, but it does mean that in effect, if you get your approval done anywhere in any state, it could potentially be marketed all over Europe. So that's a help. There's also this kind of rational and sort of clearly thought through regulatory process all the way through to marketing, and it's very much dependent on the fact that with these advanced therapies, as they call them, and they include gene therapies as well as other non-stem cell based regenerative medicine as well. What it means in effect is that if you're a patient entering a trial, then you're essentially a patient for life in terms of how you're going to be monitored by pharmacovigilance and all that sort of thing.
Some stem cells look to be immune privileged. That is, they don't engender an immune reaction, a rejection, when given to a patient from somewhere else. And if they are from the allogeneic phylum, we need to look at them very closely.
There have been a lot of claims about embryonic stem cells somehow being non-immunogenic. And what is clear is that once they differentiate, those cells become immunogenic. They're probably less immunogenic than the adult equivalent. So if you make a cardiac cell from a human embryonic stem cell and you look at its rejection qualities, it will probably be more tolerated than an adult cardiac cell taken from another human. But this is meaningless because what that means is you might reject it 2 weeks later. So it might be less immunogenic, but ultimately you get rejection. And the time spans we're talking about are relatively short. So we have to assume the problems we'll get from therapies from human embryonic stem cells will be just the same as if we took an adult cell source and I gave that to another person.
When one infuses mesenchymal stem cells systemically, either alone or in conjunction with other types of transplant, that you can suppress an immune response. Whether it does so by conferring suppression on the other cells directly or just by suppressing the immune system of the host is unclear. It's most likely suppression of the immune system of the host.
I would argue that the science is not yet at the point where I would comfortably conclude that any stem cell is immune privileged. There are certain applications where you might be able to bypass some of the more immediate immunologic problems. Retinal implantation, for instance, of retinal pigment epithelia may be one such approach. Certain applications in the brain. There may be ways of encapsulating cells so that they become, in a sense, invisible to the immune system. And once again, I think we're loath to use immune suppression when we don't have to, but if we have to, and we might, for cell transplantation, we'll do it. We'll accept the risks and complications of immunosuppression if it means a life-saving therapy.
In many cases, like spinal cord injuries, scientists have shown that you have to transplant cells into patients approximately 1 week after the initial injury. However, it takes like 3 months to prepare cells for patients by using this technology. So we cannot make it. So considering those limitations, I think another way to apply this technology into cell transplantation therapy is to generate iPS cell bank from many volunteers with various types of HLA. Instead of generating iPS cells from each patient, if we have enough number of iPS cell bank, we should be able to use those cells in the bank for each patient.
So today you've heard from our experts about the difference between autologous and allogeneic stem cells. You've heard about the differences about their safety and their universality, that is, how big a market they can reach. It's clear that they both have a place in the medical armamentarium to treat different types of diseases, and it's clear that both of them are still in their infancy and are developing under the guidance of these experts and others in the stem cell world. In the next episode, we're going to take a look at some biological stem cell processes. Differentiation, meaning sort of the downward movement from the most primitive stage to the most evolved stage. Retro and transdifferentiation, backward and across cell lineages. Migration, expansion. All of these things are going to be looked at by some of our scientific experts that are going to help you, the audience, understand the world of stem cells in more depth.
At Celgene Cellular Therapeutics, our proprietary placenta-derived stem cells hold the potential to treat a range of serious diseases. Our unique processes for differentiating and producing treatments from this rich source of stem cells will enable us to deliver these therapies on a scale similar to today's biologic remedies. Proud to be at the forefront of this transformation of modern medicine, we are Celgene Cellular Therapeutics.